Krishna Marabak. Uh, Good morning, students. Today I'm going to explain the existence of DNA damage. That is the continuation of previous class. So, existence of DNA damage have four important mechanisms: base excision repair, nucleotide excision repair, mismatch repair, and strand break repairs. So in these reactions, a nucleotide segment containing a base damage, a double helix distortion or mispaired bases is replaced by the normal nucleotide sequence in a new DNA polymerase synthesis process. So all of these pathways have been characterized in both bacteria and eukaryotic organisms. So basically, these are well-known mechanisms of DNA damage, excision of DNA damage, and these are well studied in both bacterian and eukaryotic organisms. So, what is the base excision repair mechanism? BER is initiated by DNA glycosylases enzymes, which catalyzes the hydrolysis of the N-glycosidic bonds, linking particular types of chemically altered bases to the deoxyribose phosphate backbone. So what these enzymes will do is initially hydrolyze the N-glycosidic bonds, linking particular types of chemically altered bases. So what are the chemically altered bases? Chemically altered bases means, for example, if thymine is changed to some other base, other molecule, or if hypoxanthine is formed, then immediately the base excision repair will come and repair that molecule, and altered bases will be corrected, and it attaches to the deoxyribose phosphate backbone. So the DNA damage is excised as free bases, generating sites of base loss called apurinic or apyrimidic sites. So basically, I mean, apurinic, apyrimidine, it's a not purines and pyrimidines, right? AP sites. So the AP sites are substrates for AP endonucleases. And these enzymes produce incisions in duplex DNA as a result of the hydrolysis of phosphodiester bond immediately 5' to 3' to each AP site. So as you have a DNA, it has a ribose sugar phosphate backbone and nitrogen base, right? So basically, nothing will happen to the backbone, phosphate or sugar molecule. So the only change will happen in the either purines or pyrimidines. So basically, a specific exonuclease called deoxyribophosphodiesterase or DR phase. And finally, the DNA polymerase and ligase catalyze the incorporation of a specific deoxyribonucleotide into repaired site, enabling correct base pairing. So there are two enzymes mainly involved in this process, DNA polymerase and ligase. So as you know, what is the function of DNA polymerase? It synthesizes a new strand, simply. And ligase, what it will do? It will connect the two sites. I mean, it connects the two DNA strands, ligates. It is like a linker. So base excision repair, if you see the example here, you can see you have a 3' to 5' and 5' to 3' here, and heat energy will be automatically given. So for example, here cytosine will become a uracil after giving a heat. So uracil DNA glycosylase will act on it, and it will remove the uracil, right? So once the uracil is removed, the place will be empty. right? So then the nucleases start excise the sequence of that particular, and then immediately the DNA polymerase and DNA ligase will function and it will ligate it to the normal base. Now, why this piece was removed? Because DNA polymerase can be able to synthesize the same strand which is opposite to the AGT, right? So then once the DNA polymerase synthesizes the strand, the the ligase will attach to the this to it goes and stitches the ends. So this is how the base excision repair works. Very simple example and a very simple process. So only 3 enzymes are involved, 4 enzymes. Mainly in the last step anyway, DNA polymerase, DNA ligase are the very common one, and the nucleases remove, they cut the DNA. So this is a nucleotide excision repair mechanism. This mechanism used to replace regions of damaged DNA up to 30 bases in length. So common causes of such DNA damage include the UV rays and which induces the formation of cyclobutane, pyrimidine, pyrimidine dimers, and smoking, which causes the formation of benzopyrene guanine middles. Ionizing radiation, cancer, chemotherapeutic agents, and a variety of chemicals found in the environment. They cause base modification, strand breaks, cross linkage, as I told you in the previous class. They completely change the structure of DNA. And of course, ultimately, once the change happens, the protein will be non-functional or non-active. So these are repaired by a process called nucleotide excision repair. So it's a much more complex process than BER, because BER is a very simple process, base excision repair, especially in eukaryotic cells. Because in eukaryotic cells, there will be a lot of organelles present and a lot of DNA is present inside. And all the time, the replication is going to happen and the cell needs to divide. So several gene products are required in the multiple step process, during which the ordered assembly of DNA proteins provides an enzymatic complex that discriminates damaged from undamaged DNA. So now you can see a simple photographic picture of a nucleotide excision repair. So this is a 3' to 5' and 5' to 3', both two strands. So for example, there is some mismatch or damaged DNA in these 4 bases. So it has to be recognized first where the damage and it has to be rectified. So if that happens, the nucleotide excision repair system will be active and it will unwind the DNA. So now you can see here, it will unwind. Then the oligonucleotide excision repair by cutting at 2 sides. You see here, 5', no, this is the normal one, right? So there's nothing wrong with it. So this one, we have a damaged part. So it cuts the nucleotide excision repair system by using enzymes. It cuts at 2 sides, one before damage and one, like for example, same here. Its loop is formed, right? So it cuts here and it cuts here. So to make sure that the damaged DNA part will be separated from the corrected one, because these are the correct ones, right? So then once the damaged DNA is separated, degradation of mutated DNA, and it will be degraded, and resynthesis and relegation. So, I mean, this part should be degraded. because it is mutated. And of course, what will do, the nucleases will come and do the job of removing it or degrade it. Then immediately what will happen? The enzymes of DNA polymerase and DNA ligase will go into the function and it will make a new strand. So now basically you have a new strand without any mutated DNA. So in eukaryotic cells, the enzymes cut between the third to fifth phosphodiester bond and from the 3' and lesion on the 5' side. So basically, the cut is around between 21 to 25 bonds. So thus, a fragment of 27 to 29 nucleotides long is excised. So basically, this length of this is 20, approximately 27 to 29. So after the strand is removed, it should be replaced again by exact base pairing through the action of yet another same polymerase E, and then tents are joined by the existing DNA ligase. As I told you in the previous class, all the time the last step will be done by two important enzymes, DNA polymerase and DNA ligase. So one more in E. coli, there are three specific regions called UVRA, B and C, involved in a lesion recognition and endonuclease incision. So this fragment is released by UVRD helicase action and generating a gap that is finally submitted to a repair. So now you can see here damaged nucleotide, the two, and here you have sites, UVRA, UVRB, UVRC sites, and this is the enzyme that goes and binds, and the whole thing is excised, and the DNA polymerase and DNA ligase will come and make the synthesis of new bases that will be exactly opposite to the base pair, that is the 3' to 5'. So basically this red part, not base pairs, the whole this nucleus stretches the wrong one. So this here is the corrected one. So there is one more mechanism, mismatch repair. Mismatch repair corrects the errors made when DNA is copied. So when DNA is copied during replication only, DNA will be copied. For example, C could be inserted opposite an A. So what is the Chargaff's rule? Chargaff's rule is always A adenine pairs with thymine, cytosine pairs with guanine, A, T, G, C. So for example, I mean, it's a huge DNA, and when you start replicating, the basis, the two complementary strands will be formed, and again, new strands has to be synthesized. Then two different DNAs will be synthesized, and it was taken up by two different cells. So these in this difficult, complex process, a cytosine could be inserted opposite to an A, which is against the Chargaff's rule. Or the polymerase could sweep and shutter and insert 2 to 5 extra unpaired bases. So specific proteins scan the newly synthesized DNA using the adenyl methylation with the sequence of GATC sequence as the point of a reference. So the template strand is methylated and the newly synthesized strand is not. So the difference allows the repair enzymes to identify the strand that contains the errant nucleotide, which requires replacement. The methylated part has to be replaced. If a mismatch or a small loop is found, a GATC endonuclease cuts the strand bearing the mutation at a site corresponding to the sequence GATC. immediately an exonuclease that digests this strand from the GATC through a mutation, thus removing the faulty DNA. This can occur from either end if the defect is bracketed by two sequence sites. So this defect is then filled by a normal cellular enzymes according to the base pairing rules. So here we have a simple example. You see here the CH3 both are methylated, and here we have a mismatch. Single set strand cut by GATC endonuclease. So then it cuts the one strand will be cut, have a cut here. Then the defect is removed by exonuclease. And once the defect is removed, the defect repaired by polymerase, same like DNA polymerase, and relegated by linkage, as you see here. So this mechanism corrects a single mismatch base pair, example C or A rather than T or A, or short region of unpaired DNA, very short. So whereas a nucleotide excision, you have a very long stretch of nucleotides. So the defective region is recognized by an endonucleases that makes a single strand cut at an adjacent methylated sequence. So the DNA strand is removed through a mutation, replaced and relegated by DNA linkage. This is one more mismatch with the E. coli. So in E. coli, there are a lot of enzymes present in here. Here is the strand. You see here, the G is paired with T, which is opposite to the Chargaff's rule, because G always pairs with C, right? So there are three enzymes: mute S, mute L, mute H sites. These are the three proteins that are required for the recognition of the mutation and the nicking of the strand. So basically, what it does, the mute H cuts it, the DNA here, and the DNA these two will replace and exonuclease 1, they replace X is the faulty region, and the DNA polymerase 3 will make a new strand, and the DNA synthesis will, DNA ligase will be acted upon and it binds to its normal DNA structure. So repairing strands breaks. So before I go repairing the strand breaks, so you have to understand exactly the mechanism of BER nucleotide repair and mismatch repair. Okay, so in next class, probably I will talk about this one. If you have any questions, you can just send me and I will reply you. Thank you.